Institute of Science. He went to this Biocentrum to do his postdoc, first postdoc, second postdoc at Rochester. Came back here, did lots of research, got all the awards that one can possibly get in India. I'll just name a few. I don't want to embarrass him by listing all of them.
No, you'll finish the talk also.
Shanti Swarup Bhatnagar Prize, G.N. Ramachandran, J.C. Bose, so on and so forth. He's also a fellow of all the three academies, science academies in India, and also the World Academy of Science. And in between, while he's working at IISc, he took the presidentship of Jawaharlal Nehru Center for Advanced Scientific Research. So he can do multitasking, actually. He ran his lab here and ran the entire institute there.
Okay. Thank you, Umesh, for speaking too much at this moment. Actually, I didn't want to thank Umesh because I didn't want to get into RNA domain. My forte has been working with the DNA transactions. I can only thank him because initially he wanted me to give 2 talks. I said, I don't want to give any talk here because I'm a bit hesitant because the strong RNA group, I'm not so much of an RNA person, as I said. But then somehow negotiated, but I couldn't escape giving this 1 talk. So stay tuned. Also, the problem is from the time Umesh joined, I was already there, we had been having a joint lab meeting. So he knows that this work has been done in my lab and he wants to test whether I can present it, what is done quite a while ago. So stay tuned. So this is my first time I'm talking about this work from my lab. Done, actually, initial observation was way back in 1994, but we didn't get going too far for obvious reasons. I'll perhaps tell you this. We basically work on DNA topoisomerases and transcription. And as you know, transcription and topology goes hand in hand. It's a two-way street. When the transcription is going on, or so is replication, you have to have the DNA, because mainly DNA is the genetic material, though RNA is the genetic material for many organisms, many viruses, and in other forms. So the DNA gets overwound and underwound during transcription and replication. So topoisomerases work in tandem, work together to resolve these coils. Hence, we have been working on these topoisomerases and also transcription process from organism Mycobacterium tuberculosis for a long time. But today's work is more about RNA topoisomerase activity. The work was started in Tisha Baduri's time, but she just made the initial observation way back in 1994. Then Paras Jain, I asked him to continue. He did little bit work and then he went on to do something better, I think. Then Varid contributed, and so is Fulwant Tirani. And it's an unfinished work in a sense, because there is much more things can be done in this, but I am officially retired. I'm retired before him. But because he is persistent, like our organism we work with, so I am here. Right. So I don't have any more funding. This is the funding at that time from Department of Biotechnology. Right. We work on bacterial chromosome called as nucleoid. A nucleoid is compacted by the combined action of catalytic action of topoisomerases and also the small nucleoid associated proteins. The small proteins originally called as histone-like proteins because they have properties similar to histones. Now they are called NAbs. They bind, bend, writhe, twist the DNA, so compact the DNA. Along with that, a catalytic active topoisomerase, which I will describe in the next slide. These two take care of the bacterial genome compaction, and in the process, they control many of the processes, essential processes like replication, transcription, and other processes. A point I want to say right away is, in Mycobacterium tuberculosis, unlike in E. coli and most other organisms, most other bacteria of the same genome size, both are underrepresented. I have a slide about topoisomerases later, but NAPs in E. coli are about 18 or so. Here there are only 7, and a few of them are discovered from my lab. A long-standing question in my lab is how MTB manages nuclear architecture, maintains topology homeostasis, and controls the gene expression. But for today, I just added this. In addition to these proteins, essential proteins, rRNA, tRNA, and RNA processing enzymes also underrepresented in mycobacteria. Keep it in mind, because it will come back to later. Why these essential processor enzymes involved are underrepresented is a question which we can address. We have addressed it partly, but we can discuss it later. Right. So during, let us say this is a transcription, a simplistic model representation of RNA polymerase moving. There's a positive supercoils downstream and a negative supercoils upstream, and they have to be resolved. And this is resolved by different topoisomerases, as everybody would know in this room. Simply, this is done by a relaxed supercoiling of a relaxed enzyme DNA, or introduction of the relaxation into a supercoiled DNA, and the gel representing actually supercoiled DNA converted into relaxed form. Basically, the same DNA with the same size. Excuse me, with the different topological form. Right. In bacteria, you have two kinds of topoisomerases, type 1 and type 2. So is in archaea and higher organisms. But the number becomes more when you go from yeast to humans. So your type 1 enzyme and type 2 enzyme, simply because the way they act on DNA, type 1 enzyme makes a single strand nick on DNA, and type 2 enzymes make a duplex cut. And then after, once the single strand nick or duplex is made, this intact strand is passed through. In this case, duplex strand is passed through, then it's resealed. So the enzyme has a built-in nuclease and ligase activity within it. But in between these two processes, a strand passage has to take place to change the linking number of the DNA. So you have basically only two types of topoisomerases, depending on how they function. Those of you would like to have a little bit of chemistry, which will come later also, all the topoisomerases, irrespective of from which organism you're looking at, tyrosine hydroxyl group serves as a nucleophile. Hydroxyl group activates this formation of DNA strand break and concomitant covalent linkage of the tyrosine hydroxyl into the DNA. Subsequent to strand passage, the free OH targets this phosphoester bond to reseal the DNA. So the point then is, in all these cases, the built-in vulnerability of DNA cleavage, formation of the covalent complex can be targeted by chemicals, antibiotics. That's precisely the reason topoisomerases are wonder targets for antibacterial to anti-cancer therapy. We talked about the fluoroquinolones just in the previous talk. They target DNA gyrase in bacteria, and you have many drugs targeting eukaryotic topoisomerase used in the cancer therapy. We will not discuss about discovery of new molecules, which we did for mycobacterial enzymes. But for today, before coming to the RNA topoisomerase activity, let me just show you what I said in the beginning. E. coli has the luxury of having two type 1 and two type 2 enzymes, topoisomerases. And while MTB has single type 1 enzyme and single type 2 enzyme, which is a DNA gyrase. So here, in case of E. coli, so is Pseudomonas, many other bacteria, they have the enzymes which can do backup functions and also additional functions. Whereas here, only two of them have to take up all the load. The sole relaxation enzyme in MTB's topoisomerase 1, here three of them, topo 1, topo 3, and topo 4 can do DNA relaxation. And the DNA gyrase is a dedicated supercoiling enzyme. It's the only enzyme which can do supercoiling. And the daughter chromosome separation called decatenation process is usually done by topo 4 in other bacteria, but in MTB, both are done by the DNA gyrase, primarily from our lab, and other labs have shown these studies. I think I had a slide on it. Yeah, it is there. So basically, last 30 years, we've been working on both these enzymes. And DNA gyrase is a dual function enzyme, both decatenase as well as a supercoiling enzyme. And the topoisomerase is a decatenation reaction, DNA relaxation, that is the only sole relaxation, as I said. But in today's work, I am also going to discuss its RNA topoisomerase activity, RNA hydrolysis activity, and RNA processing activity. So it is a multitasking enzyme. It's a versatile polymerase. Right. So let me just see what else is. This is what happens when you start putting together the talk just before the previous talk. So sole relaxase, single topo 1 in mycobacteria, but it is very distinct from E. coli and many other bacteria. For example, of course, what we showed is it's a highly site-specific, and they have two distinct domains, N-terminal domain. I will not go into too much of biochemistry of this, because another 25 minutes or so I have to finish the talk, otherwise, because he's in a hurry to go to dinner. So, no, you can take your time. NTD, N-terminal domain, has the catalytic motifs and whatever is required for the function. Carboxy terminal domain, highly flexible, has 3 zinc fingers in case of E. coli and almost every other bacterial enzymes. Mycobacteria doesn't have the zinc fingers. It has basic amino acid stretches. Not only that, there are many other properties, and we also find it to be site-specific. That's why it's a highly processive enzyme. So this CTD is important for subsequent discussions. Keep it in mind. One thing I want to add here is that we showed that the CTD binds non-specifically in case of mycobacteria to DNA, whereas NTD is the site-specific part because our enzyme is a bit site-specific. So there are 2 DNA binding arms within the enzyme, needed, one for binding to the sequence, second one for doing strand passage to push the other strand. This is what we showed earlier, because I may not have the data for it. So for a relaxation reaction, because topoisomerase 1 does the DNA relaxation, what you have is the DNA binding is the first step, DNA cleave, then once it's bound, in our case it binds to specific place, then it cleaves the DNA, that is its job, but it is a type 1 enzyme, it has to make a single strand make. Then the intact strand is passed through, then the second strand is, the broken strand, which we call as the gate segment, is resealed after the transport of the T segment or transport segment. Once it's resealed, reregated, then it can be released. If it has to work elsewhere, if it's site specific, it will do in the same place. Excuse me. Right. So today I will speak about multitasking activity of the topoisomerase 1 and RNA topoisomerase activity, without going into the details of all the biochemistry with or the functional aspects of the DNA topoisomerase activity. And it's a close connection and how it traverses along with the RNA polymerase, etc. Right. As I said, the work started when we started. I had started my lab a couple of years before Umesh. So my third graduate student started working on Topo 1. We've been working on DNA gyrase at that time from MTB. So during the assay development, we added tRNA, because in gyrase assay we use tRNA. For the reasons I will not discuss at this moment. So when you add tRNA, what you see is there is an inhibition of DNA relaxation. It gave us a hint that Topo 1 binds to tRNA, and possibly there is some business interaction beyond just binding. That's all we knew at that time. Subsequently, several years later, Sparrows showed that Topo 1 binds to not only tRNA, binds to RNA, these agarose gels. In some places, I may not mention which gel, what kind of gel, etc. There's lots of gels I'm going to show. It's a raw data I'm going to present. Okay. So this is an agarose gel. In some cases, it's acrylamide gel. Some cases, urea page. Some non-denaturing page. And mRNA also, you see the binding. Right. So the question now is what I showed earlier. You had there is a DNA binding, DNA cleavage, DNA strand passage, DNA religation. Does it take place in RNA world? That's the next question. So you see tRNA binding both with Mycobacterium sphygmatis and Mycobacterium tuberculosis Topo 1. This is a free tRNA shift, and we have been having a joint lab meeting, and their RNA group, many times we got tRNA from them, so we could use it. And also RNA strains are from Umesh, if I don't mention later. Right. So you see the mobility shift, that means binding. Not only that, you see the tRNA cleavage by both the enzymes, both the species, sphygmatis and tuberculosis. What you see here is a covalent complex formation. You remember, after first transesterification, you need to have the tyrosine hydroxyl cleavage and that covalent complex. I will not go into non-denaturing page where you see this. Then we did the RNA immunoprecipitation to see whether it binds to different RNAs to capture mRNAs. So you find many genes, and also including tRNA, RNA, mRNA, and many of those genes names are given. These RNAs are captured by topoisomerase in RNA immunoprecipitation experiments. And indeed, Topo 1 mRNA complex again is shown here. Right. So basically, this shows that binding and cleavage are the first two steps of the DNA topoisomerase reaction, but still not complete. This is what we are shown here. Right. Then you can prepare RNA substrates in which you can have RNA rings made, catenanes made, or knots made. They're a bit tricky experiments. I have the supplementary slides at the end about how they are made. But for the time being here, what I show you is the knot and catenanes, and these are getting resolved. And if you take a mutant, tyrosine hydroxyl mutant, that is an active site, mutant is a dead topoisomerase, you don't see resolution of those ones. And in a different percentage gel, the catenases are moving up, you are getting them resolved. Right. So then you can see whether if you show the DNA nicking activity and also resolution, that's a total activity. Because you can't show the relaxation like the DNA different topological forms. But then, does it do the strand passage? There is no lab in the world, they have done actual strand passage assay, but we have this assay. Simply because we have the antibodies which bind to the enzyme, capture it in such a way that the DNA gate, the gate enzyme where for the strand passage, that is arrested, that's blocked. As a result, we can get the DNA enzyme or RNA enzyme heterocatenates. So this is a DNA strand passage assay originally developed by Warid. The antibodies are developed by Leela Ram and inhibitory antibodies. And he developed the strand passage assay. Warid Ahmad did this to show that basically with the full length, the extent of strand passage is shown here with the deletions in the 3' ends, the relative strand passage is reduced. For example, if you have only amino-terminal domain, there is hardly any strand passage, as can be seen here. Right. So same kind of, that's basically the strategy for strand passage as shown here. That is the extent of, is basically the same slide. Right. So we did the same thing with the RNA now, but in this case, obviously we use tRNA for this kind of assay, and the schematic is shown here. And here, for example, NTD alone, you don't see the strand passage, but when you delete different regions of the carboxy terminal region, then the strand passage is compromised. Then that means we have shown most of the steps, and this is a religation assay where topo 1 with the, you can get the cleavage. After cleavage, the topo 1 should hold the RNA because of the covalent complex. And if you add magnesium, it should religate. And because then this broken RNA is gone, this is the indication that it's religated. So all the catalytic steps of the topoisomerase done with the DNA is carried out for RNA as well. And this shows that CTD has a major contributor in RNA recognition, simply because the C-terminal fragments, you can make different constructs where you can see the gel shift is abolished, and especially one particular motif is removed, then the RNA binding is quite abrogated. Right. So the question then is, whatever we showed, the RNA hydrolysis, tRNA or rRNA, has it got a relevance? Does it really participate in this process? That's where we started looking at the RNA processing enzymes in E. coli. You have, I think, 12 or 13, you can correct me, but MTB and M. smegmatis has only fewer ones. It's okay. For example, RNase G is missing and a few others are missing. So 5 or 6 of them which are found in E. coli are not there. Only 6 or 7 are found in MTB and M. smegmatis. So under limiting nucleus condition, for some reason, if the RNAs are depleted, then would the topo 1 participate in RNA processing? Remind you, there is a underrepresentation of these enzymes, as I said in the beginning. So are the topoisomerases. So basically, one can isolate the RNA from wild type and conditional knockdown. If the topo 1s are topo 1 is essential in the MTB, you can't make a knockout. In the conditional knockdown, you can generate by different tricks. And then you look at the precursor RNA accumulation and the accumulation of precursor RNA in topo CKD compared to wild type. Right. Not only that, then you can see if you have RNase E knockdown strain, whether you can complement the the deficiency. So here, what I want to show you is in a topo-1 knockdown, topo-1 levels are reduced. And in RNA-seq knockdown, RNA-seq levels are reduced, but also there is a decrease in topo-1 level. The reverse doesn't happen as is shown here. So in a topo-1 knockdown strain, this is the in vivo data, northerns, a fold change in precursor accumulation is increased. Okay, is higher, much higher. Same thing in case of RNAs knockdown, precursor is accumulated, but when you complement with the topo-1, you get some rescue. And in the northern quantification, you see this. Basically, this is what I was saying. When topo-1 is complemented, this is a topo-1 normalization. Topo level itself has come down in RNA knockdown. Now there is an adequate level, so you see some complementation. So this is an in vivo support for processing of rRNA by topo-1. Right. So in vitro, you can make northerns of looking at the cleavage at the junctions at the 16S precursors or 23S precursors or the intergenic regions. And in all the cases, what you see is you see some kind of processing taking place. And importantly, in the Y339F, you don't see such reaction, showing that it is a catalytic action of the topo-1 we are talking about. And same thing you find in the tRNA, two different tRNA operons, and again you see the RNA being degraded. And in the Y339F, phenylalanine mutant of the topo-1, you don't see this happening. What it shows is topo-1 is like participating in RNA processing, but we don't have a tRNA complementation or something. So only in vitro you can see the degradation, but we don't know actually tRNA is processed. Right. So what I said then so far is topo-1 can bind to RNA, mRNA, and tRNA in vivo and in vitro, cleaves RNA and carries out strand passage followed by religation. C-terminal domain plays a crucial role in strand passage, forms 5' phosphotyrosine intermediate during catalysis, and as RR, AR residues are in CTD are involved in RNA recognition. Topo-1 can remove RNA entanglements because we showed the knots removal. And in vivo precursor accumulation and in vitro suggest a role in RNA processing. tRNA processing, I still put question mark. We don't have foolproof data for that. Right. Now the question is, all along we talk about DNA topoisomerase. But I have a slide now to show that RNA topoisomerase activity is prevalent in all three domains of life. Those work in the last 15 years, there's quite a few papers have come up. We couldn't, though he initiated the work, but we couldn't carry out that work at that time. The reason was obvious because we are busy characterizing DNA topoisomerase activity in the mycobacteria. Right. As I said in the beginning, tyrosine OH is the nucleophilic attack on phosphodiester bond involved in, it has to do that same results DNA or RNA, because RNA just now showed that data. Tyrosine OH is needed even for RNA for that attack. But then the reaction involving, I have about 10 minutes. Yeah. Reaction involving DNA topoisomerase activity involves proton relay having these three catalytic residues, histidine, aspartic, glutamate. I'll show you in the next slide. However, in case of RNA, you have 2' OH right in the close to the 3' OH. So it could contribute. 2' OH group of RNA provides additional or alternative mode of catalysis. So RNA has two ways of doing catalysis. if it is doing topoisomerase. It could do by this process. I'll show you in the next slide. So if you disrupt the proton relay, DNA topoisomerase reaction should be abolished, but RNA topoisomerase reaction should go on. I hope I made this point. So in such mutants we have, I'll show you next slide. There should be no DNA reaction, but there should be rescue of the RNA topoisomerase activity because 2' OH is still intact. It should be able to do the job. Right. So in this reaction is a bit involved. What you can see here is, as I said, there are two modes of reaction can take place with the RNA, whereas with the DNA, it's only the proton relay. Irrespective of whether of step one mode or second mode, deprotonation of the nucleophile by tyrosine hydroxyl is the common step. A nearby water activates the tyrosine hydroxyl for nucleophilic attack. And that's what you see here in this step, tyrosine hydroxyl targeting the phosphodiester bond. In the process, you have the protonation of the leaving group, that is the glutamate 21, which is in turn extracts proton through proton relay from histidine 386 and D108. As a result, DNA cleavage takes place and concomitant covalent complex. In case of the RNA, this reaction can take place, but in addition, you have the proximal 2' OH, which provides a proton for the leaving group directly. So, if you have a mutation in these residues, D108 or E21, what happens? If you have a mutation, so this is a normal reaction. You have the DNA cleavage versus RNA cleavage, as I shown earlier. In the mutant, E21 glutamine, DNA reaction is gone, but RNA cleavage is still there. Is there the same thing in D108, but there's still some residual activity. That's possibly contributed by the 2' OH activity here. It's more clear. So basically, what I said, there are two modes of catalytic reaction with the RNA topoisomerase activity. I hope I made this point clear. Right. So in the relegation, it's for the proton relay, it's a reversal of the first one, where the free 3' OH you can do the nucleophilic attack on the phosphodiester bond, and for the second one, of course, you have the proximal 2' OH RNA extracts the proton from the nucleophile directly, as seen here. So in case of mutants, then is a topo 1, you can see the reaction in E21Q or D10A, you don't see any activity. And if the catenate to knots, for example, in these mutants, you see the reduced activity. You can see simply because the mutant has these residues are, the proton relay is abolished. So for RNA hydrolysis, it could be simply protonation of the leaving group by a nearby water, which acts as a general acid, and that is shown, is typical to a RNase reactivity seen in some of the RNases, and that could account for the RNA hydrolysis activity what you have seen. But the question is, when the enzyme does DNA topoisomerization versus RNA topoisomerase, it could not be, there may be situation where only one of the activities is predominant over the other. We don't have a clear idea, but one thing we know is pH could be playing a role, intracellular pH. What we see here is that DNA-mediated reaction works at a variety of pH from 6 to 9, whereas RNA catalysis is hindered when you go beyond pH 7. So it could be relevant in the conditions because at a higher pH, you don't want RNA to be degraded or whatever it is. It's only, so you see but when exactly such a switch can take place or whether both are functional, I have no idea. So basically then, Topo1 has both the activities, RNA hydrolysis and RNA topoisomerase activity. All steps of RNA topoisomerase reaction are carried out. Two reaction paths for RNA topoisomerase activity, canonical proton relay as well as 2' hydroxyl group catalysis reaction. Active site tyrosine is absolutely required and possibly pH dependent switch for RNA topoisomerase versus hydrolysis. To remind you, Mycobacteria works in the extreme acidic pH. So the RNA topoisomerase activity may be important under those conditions. So ability to bind and cleave tRNA, RNA processing activity is a new function, and it is a single enzyme doing all these activities. The points to consider, because it's a dynamics and evolution of RNA. So the versatility of the enzyme may be an adaptive response in the absence of Topo3 and Topo4 in the Mycobacteria. That's why it is acquired RNA topoisomerase activity also. In E. coli, we know now RNA topoisomerase 3 is involved in RNA topoisomerase activity. So RNA topoisomerase activity, cleavage and hydrolysis activities of the enzyme raise the prospect of potential origin in the RNA world. Because there are several people talking about even the topoisomerase might have originally originated in the RNA world. Still debated, of course. So it's possible that an ancestral RNAs could have acquired the capability of DNA cleavage and religation via covalent catalysis, but did not lose its competence to act on RNA. That's the other possibility. Any other possibility. But the question is, if it is doing already so many things, there could be other biological functions for this enzyme. But before that, I just want to say that in the last 15 years, we know many RNA topoisomerase activities. In humans, Topo3 beta has a RNA topoisomerase activity. So it's in many other organisms. In fact, human Topo3 beta is very essential, embryonic lethal. So these are listed here. Similarly, RNA ligase, in fact, even phage T4 tail fiber has RNA ligase activity. And there are anticodon nucleases. So the nucleases in the RNA world targeting a specific region, and ligases are very there. So sometime around evolution, they have come together to make a topoisomerase function. This is the point. And there are newer ones have also arrived. So basically, then you have the single enzyme doing all the jobs, a multitasking topoisomerase. But is it all? Does it have additional function? Possibly yes, because during transcription or replication, if there is too much topological stress, there will be hypernegative supercoiling behind, and there will be also, if you put a block ahead, that also creates a very difficult situation for a traversing polymerase. Under such conditions, polymerase can backtrack, and also RNA gets extruded out, and it can form RNA-DNA hybrids, so-called R loops. In fact, R loops are the most deleterious for any genome. The question is whether R loops whether Topo1 has anything to do with the R loop removal. In fact, there is evidence about R loop removal by the E. coli Topo1 deletion leads to hypernegative supercoiled DNA and R loop formation. So is Topo1 and Topo3 inhibit R loop formation to prevent unregulated replication in chromosomal TER region. DNA Top1 differentially modulates R loops across the human genome. So we have increasing evidence that R loop removal also Topo1 could be involved. And in our conditional knockdown strain, we can see the topo depleted, and under those conditions, you see accumulation of R loops. How exactly it removes something, I need a new graduate student to work on that, because I am already officially retired. So possibly there are additional functions for this enzyme. With that, I just want to say one slide that both Umesh and I, we work on organism like tuberculosis. Is not just a lung infection, practically infects every organ in the body. So one of the reasons I have been working on topoisomerase and transcription is to find new drugs, new inhibitors, which is a parallel thing which I have not discussed. So it's every tissue it can infect. And right now with the MDR, XDR and TDR strains is very rampant. So we continue to found new molecules targeting, but then that is for another day, not for RNA biology meeting. Thank you for your attention.
Questions?
Does that imply different kinds of substrates being targeted, or do you think
No,
same
no.
RNA substrate gets affected by different modes of catalytic activity?
No, what I was saying is DNA topoisomerase reaction can go on with the three residues, histidine, aspartate, and glutamate participating in the tyrosine hydroxyl cleavage. Whereas in case of RNA catalysis, in addition to that canonical pathway, the 2' OH comes into play after first nucleophilic attack. So you have two ways of doing RNA topoisomerase reaction compared to DNA topoisomerase reaction. So if you mutate the residues involved in that one, I can go through the slide slowly with you. Still RNA topoisomerase activity takes place. At wherever the substrate, whichever the substrate.
Just extending on that question. So quite likely when you're doing this 2' hydroxyl making a nucleophilic attack, you will end up making a cyclic phosphodiester bond.
No, yes and no. Nobody has, that's a good point, but I think if it is there, we are not bothered to check that.
Yeah.
I guess in some of the nucleases, RNAs, you don't get the cyclic thing. Yeah.
Yeah, but there are different mechanisms.
Yeah,
One is
yeah,
direct,
yeah.
but when you have
Yeah,
that.
but it's possible, but I think it doesn't happen here.
Yeah.
Both DNA and RNA substrate, is there any preference?
That's a good point. This is what I asked to check the affinity. For a single strand RNA, it was done in the lab, but not to my satisfaction. And so single strand RNA and DNA, there is no difference. But however, we have a variety of substrates, right? I don't know tRNA versus mRNA. I don't know tRNA versus double-stranded DNA. So, see, this is a topo 1. Even double-stranded DNA, it melts and prefers single-stranded regions. In fact, some of our earlier work has shown how SSB stimulates topoisomerase 1 activity by melting in the local regions. So, I don't have a direct answer what is better. This is what I had wanted to do with the identical length substrates, both RNA and DNA, in vitro synthesized RNA, DNA, and then check affinities and see whether even the RNA, the same DNA has a site specificity. Do you have that kind of site specificity or not? But from the data, what you see this, I think the activity is relevant. See what's happening in Sandeep. In eukaryotic systems, there's a disease relevance has come up and they study, but the mechanistic studies have not come up with RNA topoisomerase as much what I have shown, the different steps of the reaction, etc. Yeah, okay. Well, this is something I would like to do, not done. That would help to tell us when it came first. But even if to extending a longer answer, even though it might have been originally RNA topoisomerase activity, but I think once the most of the genomes acquired the DNA as the genetic material, it was imperative that DNA topoisomerase activity had to be there to resolve the transcription nexus. Topology nexus. So
is the topo one which has that extra insert sequence in the?
no, that is
microvector?
gyrase.
That's gyrase.
Microvector gyrase, yeah. But this has a C-terminal like a basic stretch instead of zinc fingers, yeah.
That's the one which is involved in
Yeah.
binding RNA.
Yeah.
Okay. If there are no other questions, we can break for tea and poster